Yeah. Okay. So welcome. What is this? Which is this? Okay. So DNA is a genetic material. Replicate DNA should be able to replicate faithfully. This is the criteria of all genetic materials. Genetic materials criteria is important. Have a coding capacity to generate all the proteins. This is also genetic material must carry out two jobs: duplicate itself and control the development and rest of the. Okay, what I'm particularly interested in experiment by Meselson and Stahl. Yeah, this experiment. So, starting explaining this experiment, it was supposed to know what was the objective of this. Objective was to understand if DNA replication is conservative, semi-conservative, or disruptive. What are these? Will come in the next slide. So basically, what the hell? This is not coming. So conservative means that you will keep your original DNA sequences, and the templates won't share with the daughter template. So this is the old template, and this is the blue one is the new template. The semi-conservative says that you have one template of each, and the disruptive say it doesn't matter. You have like the gaps filled with extra things, right? Okay, so what the Meselson Stahl did basically was they grow the E. coli for many generation in N14 medium. So every E. coli was pure in N14 medium, right? Then after that, what they did was extract the DNA from the cell. So they again grown many another batch with many generation in N15 medium, extracted DNA from these cells, and they mapped their density point in cesium chloride. Cesium chloride, because cesium chloride is very heavy and it could distinguish between the materials with very minute density difference. So this is there, this was there, and if it is semi-conservative, then you will get a band of, let's say, N15 and a band of N14. So it should lie between the band of N14 should be higher, I believe, and N15 should be lower. So this band that they are talking about should lie somewhere here, okay? Somewhere here. So yeah, and if it is, let's say, uh, if it is conservative, then you will only see these two bands, not the third band at all. And if it is disruptive, you will see only one band, right? Only one band or a smear, basically, of bands. But so what they did was nutritional requirement: carbon, nitrogen source, phosphorus, trace elements, and oxygen. Nitrogen is supplied from outside in case of missiles. What is MNM? I don't know. Matthew Meselson with ammonium chloride that either heavy or okay. Uh, so the only nitrogen source was this ammonium chloride that had either heavy or light nitrogen in its molecular structure. This molecule will enter the cell, enter the metabolic cycle, and generate nitrogenous bases. Uh, Important for DNA, right? Next, they have to find out one generation. So basically, what happens in a cell is a cell uh, starts in a lag phase, and then it goes in a log phase where it grows exponentially, and then it stabilizes, population stabilizes. due to the depletion of resources and all. So log phase is where you could find the generation time. Generally, this log phase generation time is linear because generation itself is like double in this time. Because this is exponential, generational time will be linear basically. So season chloride gradient because it could identify between heavy and light nitrogen. That is here the case is only one nitrogen case, so it's important. So conservative is ruled out because uh, on the first application you get only one band, which is the possibility of semi-conservative or dispersive. Now on second application you will get two bands. One is of the same as band with the nitrogen of the medium, and second a band which is a hybrid of the medium nitrogen and the original medium where the bacteria were grown, and the disperser should show only one band in all the cases, but this is not the case. So this is taken semi-conservative. Isotopes of nitrogen were used. These are non-radiative. These are heavy isotopes. Only one neutron nickel. So this is N14, and that was N28 and N29 was used. Okay. Uh, so generation 0, you get a P. Generation 0.3, you will start to get another peak. Generation 0.7. Uh, this new peak, the things shift to new peaks. In generation 1, you will get only one single peak. 1.1, this, 1.5, this, 1.9, this, hybrid, and basically you'll get then 2.5, this, 3, this, 4. So the original thing will start get diminished because there is only one stand left. So more and more peaks will signify that the more and more of medium nitrogen is getting incorporated. So this is this 0 and 4.1 next. Yeah, this. Okay, fine. Bacterial DNA replication is a form of single origin replication. There is only one. So bacterial gene is like this, and it is like this. So there is full single origin of replication. Meselson and Stahl classic experiment has one slight problem. The culture they use was not synchronized. That is, not all cells are at the same stage. So how did they get this good result? What result would expect non-synchronization culture? So cells that have just started and cells that have almost finished. Black is heavy DNA and red is light DNA. So if you have something like this, so this DNA, if it is double stranded while putting on the case, so you will get something like uh, so density of N15, sorry, ah density of N29 and density of N28. Let's see what they have used. N14, N15. What the fuck? Okay, what I'm doing here? No, oh, N14, N15. N14, N15. N15 is used, so ah, N14 and N15 is used. So if let's say if there is like X amount of replication happen, so you will get 1 minus X dn of what let's say initial was 15 plus X amount of dn15, right? dn15 plus dn14 by 2, and this X is variable, sorry, and this X is variable across the board, so you will not see a very sharp band. You will not see a very sharp band, you will see a smears of band, no? This is not good. This is really not good. So this will be a mix of all kind of cell. We expect a continuous shift in density over time. Which model will the data support? How did they get a sudden shift in density? So the only thing that is there that you will kind of break these into, let's say, these kind of uh, small fragments. What the fuck? These kind of small fragments where either this is either it is a hybrid or it is a pure one. So you will get a sharp bands. You will get sharp bands across the board. Hypothesis about why Meselson Stahl obtained the result they did, sharing the bacteria chromosome into 400 base pair fragments. The average fragments had heavy and or light nitrogen. Go back to previous slides. One origin of this may have had a transition point where both heavy and light nitrogen were found, but frequency in genome is just one, so did not contribute. So if only if it uh, get sheared at the origin of replication or around the origin of replication, then there was a chance of smear. But again, there is only one origin of smear, origin of replication, so it was really difficult. And the whole thing was very less. Now, the conclusion is semi-conservative replication. Double helix needs to unwind. Each strand is a complete for new DNA synthesis. Okay. Fine, fine. So to get the generational time, get the grow doubling time, you need to see on the log phase, log lag and stationary phase. So you have to see the log phase when the bacteria are proliferating, and you will see that number is 200 to 400 in like 1 hour. So generational time is like 1 hour, right? Okay. DNA replication is bidirectional. Very so build upon our knowledge DNA replication. So we'll just stop recording here and we'll continue from